Before we will going into our labs today, we will take a rapid review of our previous lab, which was the antibiotic test and Milan test. In the antibiotic test, we will detect the presence of aromatic amino acids. The reaction involves the nitration of the benzene ring of the tyrosine or tryptophan, results in a yellow or orange colored compound. In the Milan's test, this test is specific for phenolic compound, and the phenolic amino acid tyrosine will yield a positive result. Also, the reaction will involve a nitration of the ring of the phenolic compound, and then will complex with mercury to result in a red colored compound. Hopkin Cole test is specific for a tryptophan. Tryptophan contain an indole ring, so this test is specific for an indole ring and a tryptophan. The principle of the test involves the reaction between a tryptophan and a Hopkin Cole reagents. A Hopkin Cole reagents contain a glycolic acids that will combine two molecules of a tryptophan, oxidize them, then dehydrate them to yield a violet color complex. There will be in a reagent also a sulfuric acid, which is adding to the reagents to help in stabilization of a glycolic acid and prevents its decomposition into carbon dioxide. Here is the principle of the test as mentioned earlier. There will be an oxidation reaction and a dehydration to yield a purple colored product, and the presence of sulfuric acids to help stabilize glyoxylic acid. The Hopkin Cole reagents consist of a glycolic acid and concentrated sulfuric acid. In the procedure, to a 1 ml Hopkin Cole reagent, we added a 1 ml of a protein or amino acid solution. Then carry the test tube in a sloping position. Carry the test tube in a sloping position and add 1 to 2 ml of sulfuric acid down the sides. So you should carry the test tube in a sloping position and put the sulfuric acid on the sides of the test tubes, not on the liquid directly, no, on the side of a test tube, sloping test tubes. And then you will observe the formation of a purple color pigments at the interface between the two layers. You will notice here in a positive test we have two distinct layers and a purple ring in the interface between the two layers. Why there is two distinct layers? Actually because we adding a sulfuric acid which is much heavier than water. So here is the protein solution, which is the upper layer is a protein solution, which is aqueous solution, and the lower layer is a sulfuric acid solution, which, as I mentioned, it is much heavier than water, so it will lie as a distinct layer in the distinct lower layer. And the interface between these two layers, there will be a reaction, the oxidation and dehydration reaction. We will use this test for the detection of proteins and amino acids in a sample and to identify tryptophan from other amino acids. In the Hopkin Cole test, there is limitations, that it's nitrites, chlorates, nitrates, and excess chlorides will prevent the reaction. Why? I hope you answer this question carefully. This is one of the important questions. Why these substances can prevent the reaction from occurring? I will give you a hint. These substances will react with a sulfuric acid, and you should know what's happened later. Another test we will talk about today is a Pauly test. This test is specific for tyrosine and histidine. The test was named after the German chemist Hermann Pauly, who first described it. As you see here, the tyrosine has a phenol group and the histidine has an imidazole ring. That's what will react with the Pauly's reagents. The principle of a Pauly test involves two steps actually. In the first, there will be a formation of diazonium salts. The diazonium salts will be formed by the dissolvent sulfonylic acids and hydrochloric acids in the presence of sodium nitrite. Then these diazonium salts will couple the amines, phenols, or imidazoline ring that is found in tyrosine and histidine to give us a varicolored compound called azo compounds. The reaction requires the medium to be alkaline, that's why we're adding sodium carbonate. The reagents of a Pauly test consist of sulfonylic acids that is chilled. Why? Hydrochloric acid, sodium nitrite that's chilled also, chills me cold, and 10% sodium carbonate. The first three components will give us a diazonium salts, and the diazonium salts require the medium to be cold. That is why we use it as a cold condition. In the procedure, we in a test tube added a chilled sulfonylic acid that's dissolved in hydrochloric acids, and then added few drops of sodium nitrate to what? You should answer. Then add 1 ml of amino acids and put the test tube in a mixer, which is a vortex, followed by adding few drops of sodium carbonate, and know what happened, which color will appear. You notice here a red color means the positive test, that's a histidine or tyrosine is present, and the negative, the absence of the color, means there is no histidine or tyrosine. The test uses, the Pauly's test used to detect the presence of a tyrosine and a histidine containing protein. Also, it's used to differentiate these two amino acids from other amino acids. The test limitation, as we mentioned, the test needs a cold temperature, so this is one of the limitations of this test. The second limitation is that this test does not differentiate between histidine and tyrosine, while Milan's test does, and you should know how the Milan's test will give us a positive tyrosine and negative histidine results.